This is continued from 4A lab. The purpose of this lab is to see the effect of pasteurization on raw milk. In the previous lab, we made dilution series for pasteurized milk and raw milk and plated them using pour plate technique. Now we will look at those plates and count the number of colonies on each plate. We will only count if the number of colonies is less than 300. The numbers between 30 and 300 are considered statistically significant. You can find the pictures of a set of 10 plates, 5 for raw milk and 5 for pasteurized milk, in the Quantitative Plating Lab Report module. You need to count the colonies on each plate. This will be your results for the lab report for this experiment. Now let's see how to count the colonies. Each colony is formed from a colony forming unit. So we are counting the number of colony forming units or CFUs. If the plate is covered with thousands of colonies like you see in this picture, we consider them as too numerous to count. Obviously, they have more than 300 colonies. So you can record these results as too numerous to count or TNTC. And if your plate have these like tiny speck-like things embedded within the agar, they are also colonies. So if you remember, we mixed our sample with the agar when we poured these plates and then we let it solidify. So these are colonies that are forming inside the agar layer. So they should be counted too. The number of colony forming units that you get for each dilution is the number of colony forming units present in 1 milliliter of the diluted milk sample. We want to calculate the number of colony forming units in the original undiluted milk sample. So when we made the dilution series, we took 1 milliliter from the original milk sample and diluted it through a series of dilutions and then plated either 0.1 or 1 milliliter to get the required dilutions. So we plated 10 to the power negative 3, 4, 5, 6 and 7. And now we are counting the colonies in each of these dilution plates. So all these numbers correspond to the same milk sample. Here's the formula to calculate the colony forming units per milliliter of the original undiluted sample. The CFU per milliliter of the undiluted sample equals dilution factor times number of colony forming units at each dilution. The dilution factor is the inverse of the dilution. Let's look at an example. If the number of colony forming units on the plate for the 10 to the power negative 5 dilution for a raw milk sample is 120, what is the colony forming units per milliliter of the undiluted raw milk sample? So first we will multiply the 120 by the inverse of the dilution. And the answer is 120 times 10 to the power 5. The final answer should be written in scientific notation. Scientific notation is a way of writing a very big or a very small number with a number between 1 to 10 in decimal form and multiplied by a power of 10. So our answer for this example is 1.2 times 10 to the power 7. In your lab report, this is how you should arrange your results. Make 2 tables with 3 columns, one for the raw milk and one for the pasteurized milk. First column is the dilutions. Second column is the colony forming unit counts on each plate. Third column is the calculated colony forming units per milliliter of the undiluted milk sample. You should have one example to show the steps of how to calculate the CFU per milliliter of the original sample. If you look at the CFU counts, there is a 10-fold reduction as you go down the dilution series. If your results don't follow this pattern, it is due to some kind of errors. Discussing errors is an important part of your lab report. I will explain some of the results that are caused by an error. If you look at this example table, on the 10 to the power negative 5 dilution, there's 54 colonies and in the 10 to the power negative 6 dilution, there's 125. This cannot happen because there's more colonies in the more diluted sample compared to the less diluted sample. If you use these numbers to calculate the CFU per milliliter of the undiluted sample, you will get large variations. And these numbers correspond to the same sample, so there shouldn't be this much of big variations. I will explain the reasons for this kind of errors when I explain the discussion part of your lab report. As a final result, use the CFU per milliliter that you calculated using CFU counts between 30 and 300 and calculate the average CFU per milliliter for your milk samples. I have shown an example here, and according to this example, the average CFU per milliliter is 3.87 times 10 to the power 6. These are the accepted limits for consumption of milk. The accepted limit for raw milk is 10 to the power 5 CFU per milliliter, and the accepted limit for pasteurized milk is 2 times 10 to the power 4 CFU per milliliter. So when you calculate the average CFU per milliliter for your raw milk and your pasteurized milk, you can compare the result with these accepted limits. These are some advantages and disadvantages of using quantitative plating. Advantages are it's a relatively easy, low-cost method to estimate the colony-forming units per milliliter in a microbe-dense sample. Disadvantages are it gives only a low estimation. Many organisms are not able to grow on culture media, so they will not be counted in this method. And human error and contaminations can compromise the results. So what are the advantages and disadvantages of pasteurization? Advantages are it's an easy way of making food safe for consumption. There are no high temperatures used in this method. Therefore, we kill the pathogens, but the nutrients of the food remains intact. Disadvantages are it has relatively short shelf life because the microbial spores are not killed in this method, and therefore the food needs to be refrigerated once the containers are opened, otherwise the food will spoil. Alright, this is the fun part, the lab reports. The format for the lab reports is found in your laboratory supplement, page number 6. We will have this information posted on Canvas. So read it and follow the guidelines given in this. Make sure you have the 5 mandatory sections: purpose, procedure, results, discussion and conclusion, and references. The purpose should be short, 1 or 2 sentences to explain why we're doing an experiment and what are the techniques and organisms we are using. Usually you don't need to write procedures unless otherwise specified. Results should be arranged in tables, charts or graphs, and if pictures are used, they should be explained, they should be labeled. And discussion and conclusion can be combined together, and you will be asked some questions that you need to answer in the discussion. Discussion should be written in a paragraph form, not in point form. References, you need to use referencing style. You can use whichever you want as long as you keep it consistent. The lab report should be typed, obviously, double-spaced, and 3 pages maximum without including figures and tables, except for the unknowns lab, which is a longer lab. The maximum page number is 8 for this. And bacteria species names should be italicized and written in the correct format. So for example, if you're writing Escherichia coli, Escherichia is the genus name. The first letter should be uppercase and the rest of the letters are lowercase. Coli is the species name. It should be all in lowercase. I cannot emphasize this more. Plagiarism is not tolerated. Do not copy from other people's lab reports. You can work with your friends, but lab reports should be your individual work. You can use outside sources, but don't copy word for word. Don't write within quotation marks. Use the information, write it in your own words, and cite. You can use citation method you like I mentioned before, as long as it's consistent. Lab reports are due on Canvas before the posted due date. Late lab reports will be accepted up to a week, and for each day, 10% of the marks will be deducted for a week, and after that, 0% marks will be given. So here's how you should cite if you're using outside sources. First of all, get your information from reliable sources. Wikipedia is not a reliable source. Use books, journal articles, or reputable web pages. If you're using information from the lecture notes, mention according to lectures or according to Dr. Barty. If you use information from PowerPoints, there is no need to cite. If the lab supplement is used, mention the page numbers and the chapter name in the list of references at the end. Using outside sources is not compulsory, but for some lab reports you will need to do so. So you should have both in-text references and a list of references at the end of the report. If you follow these instructions and complete the lab reports on time, you will do well and you will have a good grade. So follow the general format when you write lab reports, and these are the specific things that you need to include in your quantitative plating lab report. Write the purpose, so not more than 2 sentences. For the procedure, explain the dilution scheme. In 4A lab, Sam explained the dilution scheme in detail. Write it in your own words. In the results section, I explained how to arrange your results in 2 tables, and you should show one example calculation on how to calculate the CFU per milliliter for the undiluted milk samples. Then calculate the average CFU per milliliter for your 2 milk samples. And these are the things that you need to address in the discussion section. Why we use quantitative plating and what are the drawbacks of this technique? Does your milk meet acceptable limits for raw and pasteurized milk? Why do we pasteurize? What are the benefits of pasteurization? And based on your results, was this experiment successful? Next part of the discussion is explaining errors. If you didn't have any errors, you still need to explain possible errors and how to prevent them. So some possible errors are adding extra sample, contaminations, calculation or counting errors. Taking care to prevent contaminations and being careful during plating and counting will help to prevent errors.